How do chromosome behaviors influence genetic inheritance? I didn't know in my time, but you will. This lesson explores the mathematical framework behind inheritance patterns extending beyond simple ratios. By the end, you will predict inheritance outcomes and justify them mathematically. Allele segregation during meiosis produces predictable gamete probabilities. Homologous chromosomes separate, ensuring each gamete receives one allele. This results in a 1/2 probability for each allele in gametes. Dominance affects phenotype expression but not allele separation. Understanding this helps predict genotype ratios accurately. Probability rules translate chromosome behavior into genotype ratios. The product rule applies when two independent events must both occur. The sum rule is used for mutually exclusive events. Dominance alters phenotype ratios but not genotype ratios. Recognizing when to apply these rules is crucial. Incomplete dominance and codominance modify phenotypic expression. Incomplete dominance results in an intermediate phenotype. Codominance allows both alleles to be expressed simultaneously. Segregation still produces a 1:2:1 genotype ratio. Understanding these concepts increases phenotypic diversity comprehension. Independent assortment results from chromosome alignment in metaphase 1. Random orientation produces independent allele combinations. This leads to the classic 9:3:3:1 phenotype ratio. However, linkage can disrupt this ratio. Understanding chromosome behavior is key to predicting genetic outcomes. Linkage occurs when genes are located on the same chromosome. Linked genes tend to be inherited together. Crossing over can separate linked genes, creating recombinant chromosomes. Recombinant frequency quantifies gene distance. Understanding these processes increases insight into genetic variability. A gene may exist in more than 2 allelic forms in a population. Individuals inherit only 2 alleles, one from each parent. The ABO blood group system illustrates multiple alleles. Probability rules remain unchanged. Allele diversity increases phenotypic variation in populations. Polygenic inheritance involves multiple gene pairs controlling a trait. This results in continuous variation, such as height or skin color. Additive allele effects create multiple phenotype categories. Environmental factors can also influence these traits. Understanding polygenic traits enhances adaptability comprehension. Each reproductive event is independent. Use the product rule for AND events and the complement rule for at least one scenarios. Observed counts may deviate from expected ratios due to sampling variation. Larger samples reduce proportional deviation. Understanding probability aids in accurate genetic predictions. Integrated problems combine probability, dominance, and chromosome behavior. Identify the dominance pattern first. Use segregation to calculate allele probabilities. Confirm gene independence before applying rules. Structured reasoning mirrors real genetic analysis. Segregation produces a 1:2:1 genotypic structure. Independent assortment explains 9:3:3:1 ratios. Product and sum rules model gamete fusion. Incomplete dominance and codominance modify expression. Not segregation. Understanding these mechanisms is essential for genetic analysis. Linkage alters expected to hybrid ratios through physical proximity. Crossing over generates recombinant phenotypes. Polygenic inheritance increases phenotypic variability. Sample size affects how closely observed results match theoretical probability. These factors are crucial for understanding genetic diversity. Verify independence before multiplying probabilities. Distinguish genotype probability from phenotype probability. Recognize when ratios suggest linkage or gene interaction. Interpret deviations using biological mechanisms. This checklist ensures readiness for complex genetic scenarios. Complex problems combine simple principles. Reason from mechanism to mathematics. Avoid memorized shortcuts. Structured thinking prevents common errors. This approach mirrors real-world genetic analysis. Identify expression patterns in genetic problems, determine gametes and verify independence. Apply the correct probability rule, compare expected and observed data. This structured approach aids in solving integrated inheritance problems. Polygenic inheritance increases phenotypic variability. Additive allele effects create multiple phenotype categories. Environmental factors can modify quantitative traits. Understanding these interactions enhances adaptability comprehension. Phenotypic variability is crucial for population survival. Apply product sum and complement rules in genetic scenarios. Independent events use the product rule. At least one scenarios use the complement rule. Compare observed and expected ratios for accuracy. These rules are essential for predicting genetic outcomes. Linkage preserves allele combinations across generations. Crossing over introduces new combinations, increasing diversity. Recombinant frequency quantifies gene distance. Verify independence before applying product rules. Understanding linkage is key to genetic diversity. Mechanistic understanding of genetics aids in accurate predictions. Recognize expression patterns and gene independence. Apply probability rules correctly. Compare observed data with expectations. This approach ensures comprehensive genetic analysis. Gene interaction can alter expected ratios. Linkage, lethality, or gene interaction can suggest deviations. Verify independence before applying probability rules. Understanding these interactions is crucial for accurate predictions. Recognize when ratios indicate underlying genetic mechanisms. Identify expression patterns and determine gametes. Verify independence or linkage. Apply correct probability rules. Compare expected and observed data. This checklist ensures readiness for complex genetic analyses. Understanding genetic inheritance requires a mechanistic approach. By applying probability rules and recognizing expression patterns, you can predict outcomes accurately. This lesson equips you with the tools to navigate complex genetic scenarios.